Our brains are incredibly biased, and so is our reality. Part 1.
So how does our brain process this visual and auditory information biologically and psychologically? Light information passes the optic lens and scatters this information across the back of your eye, or retina, to be processed by the photoreceptors, or rods and cones, in your eyes and throughout your brain. That light information is then turned into electrical signals which travel through your optic nerves to your visual cortex via your hypothalamus, which then sends these input signals to your extrastriate cortex, in which it gathers information from your life experience bank, using unconscious memories or associations to create context of color, shape, form, location, and motion. Your brain processes this information and probability to create the images you see. Looking at the blue cells somewhat near number 3 on the diagram, we have the cells that process this light information. There are ganglion cells, which integrate information into the single firing rate to send to your optic nerve. Bipolar cells, which are responsible for combining information. Horizontal cells, which connect receptors, and amacrine cells, which connect like cells to like. So, for example, ganglion to ganglion and bipolar to bipolar. All of these cells and means of interpretation are consciously biased processes, meaning these processes can only pull information from your personal well of experiences or experiences witnessed by your consciousness. This means that our brains have a very difficult time processing entirely new data. Bear with me, big words for accuracy and explanation immediately after. There are many non-visual photoreceptive systems in your deep brain as well. The deep brain photoreceptors are hypothalamic and septal nuclei of the paraventricular cerebrospinal fluid contacting neuronal system already present in the lancelet and representing the most ancient type of vertebrate nerve cells, called protoneurons. CFS contacting neurons and sensory type cells sitting in the wall of the brain ventricles that send a ciliated dendritic process into the CSF. Various opsins, pay attention to that word, and other members of the phototransduction cascade have been demonstrated in telesynthetic and hypothalamic groups of these neurons. In all species examined so far, deep brain photoreceptors play a role in circadian and circannual regulation of periodic functions, such as sleep and an ability to reset yourself. Opsins are universal photoreceptor molecules of all visual systems in the animal kingdom. They can change their conformation. More on this in part 2. Like, share, and follow for more.